is about giving and simplifying the basic physiology and basic knowledge for the electrophysiologist. This is the second session, and we are talking about the physiology from the point of view of physiology. We will be covering these key concepts. As Professor Mervat introduced the topic, it's very important to understand the physiology. Your understanding of the basic and the key concepts of the physiology, you will be able to understand the arrhythmia and understand the mechanisms of the diagnosis, either on the ECG, on the non-invasive, and in the invasive EP studies. So the key concept will be the conductive system of the heart, the action potential, and the different types in the cardiomyocytes, the automaticity and other properties of the cardiac cells, and how they contribute in the different mechanisms of arrhythmia. We said apply physiology because we want to implement this knowledge in the cath lab with the patient on the exams. So each of these principles, simple basic topics, will be in an applied and practical way. More fundamental and theoretical knowledge about the physiology of the heart will be covered in the second part of this session with my dear colleague, Dr. Ghada Diab. So, starting with the conductive system of the heart. Although all cells in the heart can conduct action potential, some cells are specialized for generating and propagating electrical signals through the heart and can be referred to as the conducting cells. These will include the A node, the node, or the AV node, and the Purkinje fibers. These cells have relatively few myofilament proteins and generate only weak contractile force compared to the rest of the heart or the myocardial cells, which we can refer to as the working cells. The SA node includes the normal pacemaker cells. We call it the maestro for the heart. The cell in the AV node provides the electrical gateway between the atria and the ventricle. It originates from the SA nodes, the impulse, and goes through the right atrium and the left atrium through different tracts. To the left atrium through the famous Bachmann's bundle, and then through the right through different tracts, the anterior tract, the middle tract, and the posterior tract, or the thorax tract and the Wenckbach tract and the anterior tract, reaching the famous AV node, which, as we said, is the gateway between the atria and the ventricle. And this concept, this fact, is important, and we'll see its importance during the different maneuvers in the EP. As I said, the different concepts we will elaborate in this session will be taken in details in further sessions during this course, but the main concept or the main goal we want to illustrate here is how we use the basic concepts of physiology in our understanding and in using the different maneuvers in the EP study. The Purkinje fibers are the distribution network that runs through the subendocardial space, and these are the fastest conducting fibers in the heart. And this again is a fact that we will see important to synchronize the contraction of the heart and synchronize the electricity propagating into the different cells of the heart, and then reaching a synchronous contraction, and in the meantime protecting the heart from arrhythmias and from prematures or abnormal activity. This rapid propagation of excitation coordinates the timing of the contraction of the working myocytes or the rest of the myocardium behind the conductive system. Conductive system, the depolarization starts in the SA node, as we said, and travels, and as we all know, travels through the internodal fibers into the AV node and hence to the Purkinje fibers. But the important point here is this illustration of the different action potential of the different structures of this conducting system. Same as we have divided the cells into conductive cells and working cells or normal myocyte cells, we have also different action potential and two main categories that is important to understand: the fast and long action potential and the slow and brief action potential. First, what is an action potential? It's an electrical stimulation created by a sequence of ion flux through specialized channels in the membrane of the cardiomyocytes that lead to cardiac contraction. So, two main forces drives these ion fluxes: the chemical potential and the electrical potential. The chemical potential is the ion will move down its concentration gradient from the high concentration to the lower concentration. And the electrical potential, the ion will move away from ion or molecule of like charge. And this will generate what we call the transmembrane potential, which is an electrical potential difference, a voltage, between the inside and the outside of the cell. When there is a net movement of positive ions into a cell, the transmembrane potential becomes more positive. And when there is a net movement of positive ions out of the cell, the transmembrane potential becomes more negative. And this is regulated by the ion channels, which help maintaining ionic concentration gradients and a differential charge between the inside and the outside of the cardiomyocytes. These ion channels have important characteristics and property enabling them for their function. First, the selectivity. They are only permeable to a single ion on their physical configuration, some for the calcium, some for the potassium, some for the sodium. They are voltage gated, which means a specific transmembrane potential range is required for a particular channel to be open in configuration. At all other transmembrane potential outside this range, the channel will be closed and impermeable to ions. Therefore, specific channels open and close as the transmembrane potential changes during cell depolarization and repolarization, allowing the passage of different ions at different times. And this important property is the reason for the formation of the action potential in the way we will illustrate in a few minutes. Time dependent, some ion channels, especially the fast sodium channels, are configured to close a fraction of a second after opening. And this is important so they cannot be opened again until they reach a transmembrane potential that is sensitive and voltage gated for their opening again. This is important to prevent further excessive ion influx. So they are selective, they are voltage gated, and they are time dependent. We were saying the two main categories. We have fast, long action potential, and this is for mainly the myocytes or the normal cardiomyocytes. And we have a slow, brief action potential, which is the main characteristic of the conductive system and mainly the SA node. The first type is the fast, long action potential, which is produced in the working cells. The main characteristics are working cells do not exhibit automaticity. And that we can see it on that graph. They have a stable membrane potential during the resting action potential or the resting phase of their action potential. They have spontaneous depolarization to threshold for producing an action potential. They need a stimulus to trigger an action potential and hence electricity and contractility. This is the character of normal myocytes or working myocytes, and also of the Purkinje fibers, because we said they are very fast in their conduction. But the Purkinje fiber has some degree of automaticity than the cardiomyocytes or the normal cardiomyocytes. The action potential is initiated by a very large sodium current, and this large sodium current is illustrated large and steep influx and steep action potential in the zero phase, produced by the opening of voltage gated sodium channels, and then the depolarization produced by the opening of sodium channel stimulate the opening of L-type or long opening calcium channels, which produce a long plateau in the action potential during which calcium enters the myocyte and contribute to the contraction of the muscle. So they are characterized by this plateau for which is responsible the flux of calcium channels through the L channels or L gates. The other type is the slow but brief action potential, which is the main action potential for the conductive system, mainly the SA and the AV node. It's different in its phases. They have the cells exhibit automaticity, and this we can see it on this graph where the cell is not stable. Even on the resting phase of the action potential, there is continuous flux of ions reaching their threshold for firing an action potential, and this is the reason for their automaticity. They don't have a flat line and a stable action potential or a stable phase or constant phase of their action potential. Their membrane is never constant, and they spontaneously depolarize towards thresholds to produce an action potential. The action potentials are produced by the opening of L-type calcium channels. They lack the effect of the high influx of sodium voltage-gated channels, and hence they don't have this steep zero phase, but they have a more gradual and more slower zero phase, responsible of which is the flux of L-type calcium channel through which the calcium ions propagates. Voltage-gated sodium channels are not involved in these action potentials, as we said. Okay, starting with the typical action potential of most of the working or normal cardiomyocytes. It has 5 phases, beginning from 0 to 4. The first phase, the resting phase, or phase 4, the resting potential in a cardiomyocyte is at -90. And the responsible of this maintenance of a -90 is an influx of exflux of potassium ions out of the cell until an action potential is triggered. And we said for this action potential to take place, we need a trigger. They lack automaticity. They cannot automatically generate or go into an action potential by their own. So once neighboring cells are triggering an action potential, fast sodium channels start to open one by one, and sodium leaks into the cell, further raising the transmembrane potential. Once reaching -70, so we have an influx of sodium, it raised from -90 to -70 millivolt. The threshold potential has been reached, which is the -70, and hence this is the point at which enough fast sodium channels have been opened to generate a self-sustained inward sodium current. And here starts phase 0 depolarization, where a large influx of sodium is responsible of this steep action potential in phase 0. The large sodium current rapidly depolarizes the transmembrane into 0 and even more than 0, slightly above, for a transient period of time called the overshoot, responsible of which is the fast sodium channel. And we said they are time dependent, so it will close in a fraction of seconds, inhibiting any further influx of sodium into the cell. At -40, the calcium channel have been triggered to open gradually and cause a small but steady influx of calcium into the cell. It shooted to 0, even above 0, and then here comes the influx of potassium into the cell, which will briefly and onward will return the transmembrane potential to approximately 0 again. And the calcium flow will continue to get into the cell in a steady way, responsible of generating this plateau phase of the action potential, which is phase 1, early repolarization. Now the cell has been depolarized, starting its early repolarization. Phase 2, the plateau phase, has started with the L-type calcium channels open and the calcium influx into the cell. Now starting phase 2, where we will start the repolarization of the cell completely, and responsible of this is the potassium leaking out down its concentration gradient throughout the delayed rectifier potassium channels. So the potassium channels are again out of the cell and counteracting between the influx of the calcium and the outflux of the potassium, leading to this plateau until the calcium channels are closed and only outflux of potassium is ongoing, and again the membrane potential is dropping into its resting value at -90 at the end of phase 3, which is a complete repolarization, and again into the resting phase, which is phase 4. The action potential is at -90. The constant small and leak of potassium maintaining this -90 millivolt of potential, and ready for a further stimulation from neighboring cells to trigger another action potential, and so on. So this is the normal or the typical action potential of the cardiomyocytes that are not able for automaticity. But how the different cells are communicating together and very fast synchronizing this action potential between the cells, it's through the gap junctions. So by the influx of sodium from one cell, some is communicating through the gap junction and leaking into the other cell, and so on until the whole myocardium is depolarized and repolarized again. And you can see again here the different steps. Mainly an influx of sodium is responsible for the phase 0, and then the plateau with the calcium, and then some repolarization and equilibrium with the calcium through the potassium, and then the potassium is all alone reaching again the repolarizing phase and the resting phase for the myocardium. Yet for the pacemaker cells, for the brief and slow action potential, it's a little bit different. And we will see it in this illustration. First, the phase 0, these cells do not have voltage-gated sodium channels, as we said. This is characteristic for the SA node and the AV node. They have this continuous flow of sodium through the what we call the funny channels, or the funny current. So in phase 0, there are no voltage-gated sodium channel activity, but the L-type calcium channel turn on more slowly with depolarization. This produces a slower upstroke of the action potential. So the difference here, that the upstroke or the phase 0 is only maintained and mediated by the influx of the calcium ions through the L-type calcium channel. And that's why it's less steeper than the action potential in the other myocytes. It lacks the plateau. Phase 1 and 2 are not really developed in these cells. And phase 3, repolarization, is maintained by the opening of voltage-gated potassium channels, same as in the other cells. And then phase 4, the membrane potential is not stable. Line is not flat. There is spontaneous depolarization of the membrane potential toward the threshold for producing another action potential by another opening of L-type calcium channels. And the responsible of this instability of the membrane is this passage from the funny current. During this phase of 4, there is leak of sodium ions to enter the cell, which is called the funny current and is labeled as IF. The leakage of sodium into the cell through the funny channels gradually depolarize the membrane potential to the threshold for opening L-type calcium channels, and hence another action potential is triggered, and so on. And this is the primary determinant of automaticity and heart rate. And this is how pacemaker cells make the transition during phase 4 back to phase 0, and so on. The larger the funny current, the faster the rate of spontaneous depolarization during phase 4. The smaller the funny current, the smaller the leakage of sodium during this phase, the slower the rate, the less is the automaticity of the cell. And this will explain to you how the vagal tone and the sympathetic stimulation and activation controls and increase and decrease the heart rate. Since the funny current is increased by an activation of adenyl cyclase, or by increasing the binding of cyclic AMP to funny channels, or by phosphorylation of funny channels by protein kinase A. And we know that beta-1 adrenoreceptor stimulation by norepinephrine or epinephrine will activate the J-stimulating regulating proteins, which will increase the cyclic AMP and hence will activate protein kinase A and hence increase the funny current and speeds up the heart rate. While alternatively, muscarinic M2 receptor stimulation by acetylcholine will activate the inhibitory J-regulating protein, which decrease the CAMP, decrease activation of protein kinase A, decrease the funny current, and slows the heart rate. So this is one mechanism by which the heart is regulated. For the chronotropic characteristic of the heart are regulated. Another related mechanism controlling heart rate is the increased influx of calcium through the L-type calcium channels produced when these channels are phosphorylated by the protein kinase A. And from this automaticity, as we all know, we have the sinoatrial node having an inherent rate of 60 up to 100 beats per minute. The atrioventricular node has an inherent rate of 40 to 60. And the Purkinje fibers, as we said, have their own automaticity, though they have a fast action potential with the rate inherent of 20 to 40 beats per minute. So automaticity in the cardiac pacemakers, unlike other cardiomyocytes, they do not require external stimulation to initiate their action potential. They are capable of self-initiation of the depolarization in a rhythmic fashion. This property is also known as automaticity, whereby undergoes spontaneous depolarization and action potential is triggered when threshold voltage is reached. Unstable membrane potential, as we said, they have always the instability for their action or for their transmembrane potential. No rapid depolarization phase due to the lack of voltage-gated sodium channels or weak sodium channel voltage-gated sodium channels, and in the meantime they also have fewer inward rectifier potassium channels. And we said these potassium flow is important to maintain the transmembrane potential to -90. They never reach -90 to open the voltage-gated sodium channels. They never go lower than -60 millivolt. And hence, the phase 0 is only mediated through the calcium influx, and there is no fast inward of sodium through the voltage-gated sodium channels. Finally, with this diaphragm and with this diagram showing the difference between the phases, here we have the fast action potential with 5 well-developed phases of action potential. The phase 0 is mainly mediated by the inward sodium influx. Phase 1, potassium outflux. Phase 2, calcium and doing the plateau phase with the influx of the potassium. And finally, repolarization through the potassium outflux and maintained at a very slow, as we can see, the arrow is narrow and very fine, maintaining a transmembrane potential of -90. While on the slow action potential of the conducting system or the orchestra of the heart, or the SE node and the AV node, we have only three well-developed phases. The first, the influx of the calcium in phase 0, which is less steep than what we saw with the influx of sodium and potassium and the calcium. Phase 3, repolarization through the potassium outflux, and finally the funny current, or the sodium continuous and slow influx, that raising the transmembrane potential up to a threshold for automaticity and further action potential triggered automatically. Okay, so we talked about automaticity and how the different cells have inherent heart rate from the normal SE node if backed up with a slower rate, backup rate by the AV node, and finally the last escape rhythm, as we call it, from his Purkinje fibers with a rate between 20 and 40. And this is an important protective mechanism inherent in our hearts. Healthy working cells do not demonstrate